It's very nice to meet you all. My name is Romina, and I am a grade 12 student at All Saints Catholic High School here in Ottawa. And I started my research in Dr. Lorimer's lab at the Center for Cancer Therapeutics of the Ottawa Hospital Research Institute in October. And my main area of interest is telomerase. How I became interested in this enzyme is because I'd heard of two companies who are actually claiming that based on a person's telomere length measurements, they're able to predict your physiological age. So I thought that was really interesting, so I did some more research to find out what these telomeres actually are. It turns out telomeres are these short tandem repeats of DNA at the end of your chromosomes that protect your chromosomes during replication by preventing them from fusing together. In humans, this tandem repeat is TTAGGG. As a cell undergoes the process of mitosis and replication, these telomeres start to become shorter and shorter until the defensive mechanism of the cell recognizes the DNA as damaged and not suitable for replication. And why these telomeres actually become shorter is because the enzyme responsible for DNA replication, DNA polymerase, is not efficient in replicating these ends. And so therefore, the cell then starts to undergo the process of aging or senescence. However, in highly proliferative cells, such as ES cells, there exists an enzyme called telomerase. And what telomerase is actually able to do, it's shown here, is actually able to go to the end of these chromosomes and add certain bases in. And by doing that, it protects the integrity of the telomeres. And it helps the cell maintain a survival rate. Telomerase is not activated in differentiated cells or in adult cells. However, it's highly active in cancer cells. So here we can see that in adult stem cells, the telomere is very short. However, in embryonic stem cells, this is where the telomeres are located, and telomerase is active, so the telomere length is very long. In the field of cancer research, there's a lot of interest in learning about telomerase, and there's a lot of interest in seeing if there's potential for cancer therapy through inhibition. A recent study showed that through the knockdown of a gene called hypoxia-induced factor 1 alpha, we'll just call it HIF1 alpha, through the knockdown of this gene, there was a downregulation in the expression of telomerase and in the length of telomeres in the mouse ES cells that were tested. So just to give a brief explanation of what they did in this paper, they took mouse ES cells that expressed a green fluorescence protein, and they used RNAi inhibitors or silencers against around 3,000 different transcription factors, and they were able to see which gene was actually knocked down. And with the knockdown of HIF1 alpha, they saw a downregulation in telomerase and the length of telomeres. What HIF1 alpha actually is it's used by the cells as a defensive mechanism for when they're under hypoxic conditions, which is low oxygen levels. However, in cancer cells, it's expressed even under normoxic conditions. So there has been research done for using HIF1 alpha inhibition as a potential cancer therapy, but its underlying mechanism has not yet been thoroughly explained. By reading a little bit more and looking at a couple more papers, I found this drug called echinomycin. The echinomycin is actually a peptide from the antibiotics group, and what it actually does is it inhibits HIF1 alpha activity. Echinomycin used to be used around 20 years ago in clinical studies, and it actually was used for cancer therapy. However, because of the abnormalities and side effects that this drug caused, the research was not continued. However, last year there was a paper that showed that when low doses of echinomycin, when it was used on mouse cells, there was actually positive response to therapy for the mice that had leukemia or lymphoma, which are these blood cancers. So how echinomycin actually works is that I represented it by this yellow triangle, and it binds to the promoter region for HIF1 alpha in a sequence-specific manner, and it blocks HIF1 alpha from binding to the promoter, and therefore all the target genes which are under HIF1 alpha control, their transcription is blocked. So by knowing that cancer cells, what happens to cells when they become cancers is there's a mutation. This mutation could be in the oncogene, or it could also be in the tumor repressor gene. However, in either case, with this mutation, there is an upregulation in the level of expression for HIF-1 alpha. And what my hypothesis was that the suppressive effect of HIF-1 alpha could potentially be linked to the activation of, or to the inactivation of telomerase in human cancers. Now, in order to do my experiment, I first needed to find a cell line that expressed a quantifiable level of hTERT, which is the enzymatic portion for human telomerase. And to do that, I had a number of different cell lines available in our lab, and by isolating RNA and synthesizing cDNA, I ran a real-time PCR. In my real-time PCR, there was the results you can see here. I compared CT values. What CT values actually are is it stands for threshold cycle. So it's the point where you have enough fluorescence signal for your where you have enough PCR product for your fluorescence to pass the threshold. So therefore, in this graph, it shows the lower your CT value is, the more abundant your template is in your sample. So looking at the different CT values, MCF7 had the lowest CT, so therefore it expressed the highest level of hTERT, which is why I chose this cell line for my further investigations. And MCF7 is actually a breast cancer cell line. The next step to my experiment was to find a normalizing gene or housekeeping gene. A housekeeping gene is a gene that doesn't alter at different time points, and it helps us use it as a control. And to find a housekeeping gene, I tested a number of different genes available in our lab on the MCF7 cell lines through real-time PCR once again. And I tried to look for a gene that amplified in the closest region to hTERT. And we can see here that PBGD was the gene that amplified in the closest range, and that's the gene I chose for my normalizing gene. The next step, the next step was to actually treat the cells with the drug. I chose a 10 nanomolar concentration of echinomycin because that's a concentration that's been known to completely inhibit HIF-1 alpha activity. So I treated the cells with the drug. Here I have untreated cells, and using a trypan blue exclusion dye method, I looked at the viability of these cells. So at the untreated level, you have about 88% viability in these breast cancer cells. However, as with prolonged exposure to the drug, your viability actually is decreased, where in 72 hours, you have about 25% viability for these cells. Then using the cDNA from these results, I did a real-time PCR once again. And this time, I used a relative quantification method, which is the delta-delta CT method, to compare the level of expression of hTERT at different time points. So here I can see that at the 24-hour time point, I have about 55% inhibition for telomerase. Now, in order to just clarify my data at the protein level, I decided to do a Western blot. And my Western blot did actually support my results from the PCR by showing that here in lane 2, where you have 24-hour treatment, you had the least level of protein expressed in the MCF7s. So in conclusion, my experiment showed that MCF7 is a suitable model to study the effect of echinomycin inhibition, the effect of HIF-1 alpha inhibition through echinomycin, and I also showed a correlation between HIF-1 alpha inhibition and downregulation in hTERT, where in around 24-hour treatment, you have about 55% inhibition. My results are very preliminary, but they do give us an insight of how this drug actually works, and it could help us in the future by reducing the side effects of this drug and making it more applicable for patients with cancer. Thank you.
Thank you very much, Romina. Which grade are you in?
In grade 12.
And is this the first or second year that you've been involved in this research?
I was involved in Sanofi in grade 9 and 10, and I didn't do it in grade 11, and I came back in grade 12.
Okay, thank you. Questions? Marshall?
Hi. So on your graph of the relative hTERT expressions, I noticed that it looks like for between the hours, your error bars overlap pretty much. So is there like a significance? Have you tested the significance of the different time frames? Is that important?
Like the different error bars that you see?
Yeah.
So because of the time constraint that I was put in, I couldn't do the experiment as many times as I'd like to. So that's why the error bars are pretty high in our results. But yeah, if I did have more time, I'd do more, like a number of more times, and that could lead us to having more accurate results. But in the graph, you can actually see a sudden downregulation at the 24-hour point, and then the H-shirt level actually starts to increase at the 48-hour point. And that's because I think cancer cells are really smart. So I think at the 24-hour, they start to slowly adapt to the situation that they're put in, and that's why you see an upregulation in H-shirt. But at the 72-hour point, they're not able to sustain this, this adaptability that they were able to show at the 48-hour point.
Ron?
So you said you were involved in the competition in grades 9 and 10, and then how does what you're doing now relate to what you did in grades 9 and 10? Is this a continuation? Have you been thinking about cancer, telomerase, oxygen effects, or is this a completely new approach that you've come up with?
It's a completely new approach. In the years prior, I was working on completely different projects. Like one year I did muscle tissues, and the year before I looked at cholesterol levels. So this year it's a completely different project, but I would say it's my favorite so far.
So with some of the technical things that you've done, like the RT-qPCR things, were you able to do all of that on your own, sort of be shown at once and then use the instrumentation yourself?
Yes,
So
I have my lab book here if you'd like to look at any of the methods. And yeah, I did all my experiments on my own. The members in the lab group were able to show me, and I did the rest on by myself. I would say the hardest experiment though was the Western blot.
Yes. Can I just one more sort of? It's a more general or more generic question about telomerase and telomere length associated with disease like cancer and also with aging. There's some controversy to those hypotheses, and you had mentioned that in your introduction. Can you maybe just quite briefly tell us what your sense of that is from you having done this project, any reading that you've done? Are you convinced that telomere length is associated with some of these things, or do you think it's still a little bit of an if?
It's definitely new research that's being done on telomeres. It's something that's just starting to interest researchers, but I do think it has a lot of potential in the cancer therapy or in cancer research. But I mean, if you were to use telomere measurements as a way to, there could be a lot of controversies against that, and you can see that because you could use telomeres measurements, and based on that, you could discriminate against a person based on their physiological age. So, but in the field for cancer research, I think there's a lot of potential in using it to decrease cell survival rate, which is key in cancer.
Yes, I'd have a question on the specificity of actinomycin. So your hypothesis is, or at least your conclusion is that it's the inhibition of the HIF-1 alpha activity which is responsible for the decrease in expression of the telomerase. How do you know that it's truly the case? Couldn't there be other effects of the drug that you actually aren't monitoring that could also play a role in this decrease?
Yes. So in my experiment, I didn't actually test to see if HIF-1 alpha was inhibited. I went by the assumption that these papers show that HIF-1 alpha, that actinomycin completely inhibits it at this dose, at the 10 nanomolar concentration. But if I were to test, and this is it's a really good idea, if I were to continue with this and test to see if HIF-1 alpha is inhibited, I would probably choose a target gene that's already been known to be under HIF-1 alpha activity. And I know there's a gene called VGF, which is a target gene of HIF-1 alpha, and it's known to be under the control of HIF-1 alpha. So that would be a way I could see it. I could use it as a control for achinomycin.
How long have you been working on this project?
I started in October.
Just in October.
Yes.
Okay, and the acinomycin you mentioned, did you find it yourself in the literature, or was it suggested to you that that would be
we
a
were-
potential inhibitor?
Yeah, we were looking at different ways to inhibit HIF-1 alpha with my mentor. And at first we wanted to use the knockdown, like gene-specific method, but that was really hard in the time frame that we had. So my mentor found this article for acinomycin, and that's how we became involved by using the drug. And it's really interesting because I think that knowing the underlying mechanism of these different genes and these different drugs could actually help us develop better therapies, because we know that a drug inhibits this gene at this level, and we see a response down here. However, there's a number of different pathways that this drug can take, and there's a number of different side effects that could be caused with this drug. And by knowing the mechanism, then we could minimize these side effects and we could develop better therapies. Yeah.
Actually, that almost answers my question, but leads into the question about the side effects. So one of the things you said at the end is if we know these things, then perhaps this is a drug that could still be useful. Do the side effects, and I don't know what they are, give you any indication of the types of pathways that this drug might have affected before? Would it have been related to this? And even if you know the mechanism, does it still make it a useful drug, given depending on what the side effects were? As I say, I'm not sure.
The side effects were actually kidney abnormalities that were caused, and I haven't looked in depth into the side effects to see how we could minimize it yet, because this is very basic research at this point. But yeah, that could be a different step, and in order to give it some commercialization value, you do need to finish your results and show a direct link between HTERT and HIF-1 alpha, and then once that's done, you do clinical studies, and that's where you could look at the side effects and abnormalities that are caused by the drug. Does that answer your question?
So we have examples of some products that at high levels are very toxic, and at low levels can actually have commercial value, like Botox, right? Do you see this for the acinomycin?
I do, because when it was used 20 years ago, it was used at very high concentration. So maybe that's why that you were seeing these side effects in these patients. But at low concentrations, they showed that in mice, it actually it helped with the treatment of leukemia and lymphoma. So that could be a potential for this drug.
Let's assume you continue the great work you started and you continue to show good results. Who would you approach moving forwards for your clinical studies and so on and so forth?
Like what group of, like which population I would target? I haven't looked into it. I'm not sure how they go about doing clinical studies, but I would assume there's different tests that need to be done to make sure that the patients are healthy and they can do the testings, or well, actually to see if the patients have different cancers. And like you could look at, I only looked at breast cancers, but you could do this with different different types of cancer and see if you see the same link.
Who manufactures this drug?
I believe it was purchased from Sigma for us, but I'm sure there are other companies who are making it. And it's actually very, it's not very expensive, so it's very economic. It's an economic choice.
Ron, the last question.
Yeah, just briefly, one thing. In your earlier experiments, when you tested various controls to see which was the closest in your gene expression studies, you decided that there was one, and I can't remember what it was. Yet when you did your Western, so PBGD, and then you did your Western, your control was what most people use, and that's GAPDH.
Yes.
Why the difference? And do you think that might have been worthwhile to use the same gene as a control in both of those experiments?
Yes, that would be a good option, but we had GAD-PH antibody available in our lab, so that's partly the reason I chose to use it in my Western.
Any other question? If not, thank you very much for being here. Enjoy the rest of the day.
Thank you.